All right, let's humble ourselves and pray. Almighty God, thank you for today. Thank you for this nice day in Uganda. Lord, as we are going to have this lesson, I want to call upon your Holy Spirit to come and be with us. Please help us, Lord, to understand. Pray that each and everything goes well, and also our connection, that it may not disturb us. Also pray that we may be orderly in the class and not make noise. And above all, God, may your will be done. In Jesus' mighty name I've prayed. Amen.
Amen. Thank you, Patricia. Yeah, so and also one other thing, I would like to request us not to put our videos because one, they will disrupt the rest of us, but two, they will consume much of your data, yet you need that data for other days and other things. Okay, so I'm kicking off from where teacher Silverine stopped last week. Teacher Silverine was able to take you through some parts of respiration. And so I'm going to continue from there. So I'm hoping that most of us attended that lesson and we can build on that. Otherwise, it's not possible for us to go back and reteach what was covered in the interest of time and also in order to respect those who attended. So we are going to continue from where we stopped. So I'll be sharing my slides in a short while.
All right.
So I hope we can all see my screen. So right now it is blank. And on the screen, just as a quick reminder, we looked at the steps that are very key when we are constructing or designing an experiment. Now remember that in our new curriculum, which you have been dealing with now for the last maybe 2 years, we are expected to design our own experiments. And as you design an experiment, there are certain procedures you must follow or certain things you must bear in mind. And so one of such things is to ensure that you know what you want to do. So it's like you want to build a house, you must know what you want to do or what kind of house you want to build. So what is the objective? What experiment do you want to conduct? Do you want to test a certain substance for the presence of starch? That would be like an aim or the objective. Do you want to show that during respiration, a certain product is given off or a certain raw material is necessary? So that is your objective. And then you come to the hypothesis where we say that now you can say that such and such is necessary for this process to happen. For instance, you could say oxygen is needed for aerobic respiration, or you could say oxygen is not needed for aerobic respiration. So the hypothesis is usually either a positive answer or a negative, rather a positive statement or a negative statement. So I'm sure as you have been doing this at school, you have been taught about this. So the statement is either a positive one or a negative one, but at the end of the day, you want to prove whether that statement is correct or not by conducting the experiment, okay? So therefore, if you're going to conduct an experiment, then you must know what you need to use. So what you will use is determined by what you want to do, okay? What you'll use is determined by what you want to do. So you have to take time and think through what might be necessary. Especially, it's important that your experiment is done with available materials, okay? With the available materials, because there can be certain options, but when they are not cheap, okay? So we encourage you to design experiments within the available materials, because you don't have the luxury of lots of resources to have the most elaborate experiments. Otherwise, you're supposed to work with what you have. And then you must know the step-by-step process of putting these materials together in order for you to conduct the experiment. So while you're conducting experiment, you will find that there are certain issues to do with safety, and you must take care of those, because the experimenter and other people must be safe. Otherwise, then science becomes very dangerous. So, but as scientists, we must look at safety. So the safety can be in terms of accidents, or in terms of maybe exposure to microorganisms that could cause disease, or any other thing that is unsafe. So you look for ways of removing the unsafe practice, or making the whole thing safe. For example, if you're dealing with hot things, how do you ensure that you're not burnt, and so on and so forth? And then errors, you must look for ways of ensuring that there is minimal error in your setup, so that your experiment works out, or is able to answer the question, or be able to really work, if I could put it that way. Then also you write the observations when you conduct the experiment. Now, expected observation I wrote here was simply because if you are doing it in an exam, then you would write the expected observation, because you're not actually carrying out the experiment, you're just saying what would be expected. But if you're doing an actual experiment in class, then you write what you have seen, okay? You write what you have seen, and then from what you have seen, then you can find a way of explaining it. You analyze that observation or those results. And then once you have analyzed them, then you can now conclude. So your conclusion is either going to support your early hypothesis, because you started with a hypothesis, and then you want to see whether that hypothesis has been confirmed, or it has been rejected, okay? Because, for instance, if you said that oxygen, okay, let me just say carbon dioxide is necessary for photosynthesis, and then the experiment shows otherwise, then you would have to say, therefore, carbon dioxide is not necessary for photosynthesis. But if it shows that, then you can conclude and say carbon dioxide is necessary for photosynthesis. Now, what happens is that sometimes, because of errors, the thing may not work out. So sometimes you may have to go backwards and try to see why has it not worked out, and then you adjust, okay? So that is how scientists do things, okay? That is how scientists do things. So that is just a recap of what was the last slide of last lesson. So today we are going to look at some different experiments under this topic of respiration. We are expected to design experiments in this topic to be able to show, one, that energy is given off during a respiration, but 2, also to show that oxygen is needed during aerobic respiration, and 3, to show that carbon dioxide is given off during aerobic respiration and anaerobic respiration. Okay, so those are some of the experiments you're supposed to conduct. Maybe another one is to show that water is given off during the process of respiration. Because if you recall very well that when respiration takes place, there are certain raw materials, or rather substrates that are worked on, and then the products that are produced thereafter. So maybe very quickly, we can remind ourselves about the products of aerobic respiration and those of anaerobic respiration. So let's start with aerobic respiration. Who wants to tell us what happens during aerobic respiration? Yes, Juliet.
Aerobic respiration is a type of respiration that requires oxygen to take place.
Yes, you have defined it, but I want you to tell me what is given off when this respiration takes place. Perhaps you can also tell me the reactants and then the products.
I think in aerobic respiration, it requires oxygen and it gives off carbon dioxide.
Okay, thank you. Juliet, who wants to add on to Juliet's answer? I feel it is not complete. She has not fully answered my question. Someone is, please, after talking, you mute. We don't want to participate in the conversations in the background. Let's hear from Mulungi Favor.
Aerobic respiration requires, food is burned down in the presence of oxygen, and then the products given off are carbon dioxide and water.
Okay. Is that all? Let's hear from Patricia.
Okay, thank you. So you asked for the reactants. These are it's oxygen and food. And then the products are carbon dioxide, water, and energy mainly.
Okay, thank you. So the product also includes energy. Now, what is the major food source used in this process of respiration? Because food can be very broad. I remember teacher Silverin emphasizing that point in her discussion. Naomi? Anna, please, where possible, you can interrupt and read what is in the chat.
Right, teacher.
Okay.
In the chat right now, they have written that respiration uses oxygen and glucose to give carbon dioxide, water and energy. Then the products of aerobic respiration are carbon dioxide and water.
Okay.
Yeah.
Now, the question I had asked as well was what kind of food is used in this process of respiration? What is that food?
In the chat, there are carbohydrates.
Okay. Yeah, so I remember teacher Silverin emphasizing the point that when we eat carbohydrates, they are digested, and the final products of digestion of carbohydrates are glucose. Mulungi Favor has put it there. So glucose is absorbed into the bloodstream, and then it is used by the cells to carry out respiration. So when you eat carbohydrates, the final food that is respired is the glucose. Okay, now let's talk about the anaerobic very quickly because it's something that was already covered from which we need to build. So anaerobic respiration. Do you have anything from the chat or from?
Yeah, Patricia is raising her hand.
Okay, Patricia, please speak.
Okay, anaerobic respiration. Here, like food is broken down to release energy, but it does not require oxygen. So here, like the glucose is not completely broken down, so less energy is released. But okay, the respiration depends on the type of animal, like the products which are released depends on the type of organism. For example, in plants and fungi, glucose is broken down to give us ethanol, carbon dioxide, and energy, while in animals, glucose is broken down to give us lactic acid and energy.
Okay, I think Patricia was very attentive that time. Thank you, Patricia. Okay, so we are going to build on that in our lesson for today. So we are supposed to design an experiment to actually show that energy is given during respiration. So if I may ask, what is energy? How can we show that really energy is given off? How can we prove that experimentally? Because every product must be verifiable. If I say carbon dioxide is given off, I should be able to collect it and show that what I have collected is carbon dioxide. And if oxygen is given off, then I should be able to also show. Now, when energy is given off, how do I show that it is given off? Because when we talk about energy, what do we mean? How can we prove this? Can we have some people tell us what energy is and how we can show it? Because rightly, everybody say that respiration results in the formation of energy. Amara, unmute and contribute.
Yes, teacher, good afternoon.
Good afternoon.
Teacher, I would say energy is like the ability to either lift or do certain types of work.
Okay, that's a definition. Now, in our context, in our experiment,
yes,
are we going to see the organisms lifting things, doing work? How do we prove that energy is given off?
Teacher, I would say because now doing respiration, as we say, energy is a byproduct, how we can say energy is being given off, like if they are sweating.
Okay, now sweating is limited to organisms such as you and me, organisms that have sweat glands. Now, if you are going to do this practically, we need to get things that we can put under certain controlled conditions and run experiments on them. Remember, experimenting on human beings has ethical issues.
Yes.
So we cannot easily work with human beings. We have to work with other organisms where there are no ethical issues.
Yes.
So in light of that, are there different forms of energy that you're aware of? If you know them, what are those different forms of energy?
Teacher, the different forms of energy I can give heat energy.
Mm-hmm.
Don't know if it's
The question is to everyone. So, yes.
okay.
But
So,
since you're the one on having the mic, you can talk. Then others will come in when you're done.
okay. Teacher, I'm going to only give like 3 answers, 3 or 2. One of them I have is heat energy, chemical energy, and sound energy.
Okay. Schneider, thank you. Schneider.
Solar energy.
Solar energy, other forms of energy. Will Faith? Will Faith, will you unmute? Okay, I think. Yes? Okay, let's hear from Alan.
Light energy.
Light energy. Okay, so maybe with that, we can see that there are many forms of energy, okay? But now the question is, which of those forms of energy that you have discussed can be verified during respiration? Can we show that light energy is there, or sound energy, or chemical energy? Which are those that we can prove in an experiment? And are all those forms of energy produced anyway when respiration takes place? Will Faith?
So I think chemical energy. Yeah, since respiration, because when you look at the definition of respiration, it is a chemical process. So that's why I think chemical energy can also work.
Okay. It is true chemical energy is produced, but is it possible for you to prove it in a lab, a simple lab like that one at school? To what extent can you prove that chemical energy has been produced? Are there other forms of energy other than chemical energy that are produced that we can verify? I see the chat is very busy. Thank you. Seeing Jolene say maybe heat energy. Okay. Jolene, how do we prove that heat energy is given off? Is there a way of verifying that heat is given off?
It is in glass.
You mean you can see heat in glass? Please try to tell your siblings to keep quiet, or they go and play from elsewhere so that they don't interrupt us. So Jolene has told us about heat energy. And you notice that when you are respiring, your body tends to produce a lot of heat. And so how do we prove that heat energy is given off? What's the most simple way to show that actually heat is being given off? Juliet Gabriel, do you want to answer? In the chat, do we have anything, Hannah? I hope people concentrate on what's at hand. Now let me ask, if you are having a fever and you go to a health center, how do they prove that you are having a fever? What are some of the signs that you have a fever? And how does the health specialist prove that you have a fever?
They check your temperature with a thermometer.
So when they check your temperature, what are they trying to discover? Yes, Bridget.
Okay,
Bridget?
teacher.
Yes.
In most cases, when you have a fever, you have a higher temperature than the normal one. So when they check your temperature, they're trying to prove whether your temperature has actually gone high and you have a fever.
Okay, if you don't have a thermometer, what is the easiest way of knowing that someone has high temperature?
They just touch you and see.
So when you touch the person, what are you trying to do? What are you trying to arrive at? It is true they will touch you.
Mm-hmm.
So someone says touch the forehead, then what?
And if you touch the forehead, then you will feel that the heat coming from the body is actually too much from the normal one.
Okay, that's what I wanted. So heat is actually one of the forms of energy, okay? And when something is producing much energy, it usually gives off much heat, okay? Usually gives off much heat, and the more heat produced, the more energy generated. Are we together? Yeah, so I think we have answered some of these questions. How can you demonstrate that energy is given off during respiration? What are the different forms of energy? How can you measure the energy given off? So can we answer these ones very quickly before I can show you what answers I have? Now, some people are busy disrupting us. Hannah, I give you the powers to remove those people from this room, exit them from the call. Okay, I'm going to put a waiting room where some of you are going to be put who are misbehaving. Okay, so that we don't have disruptors. So those who are annotating, please stop it. Okay, so let us answer these questions. Hannah, allow the people who are joining us to enter the room, as long as they are not those that are causing problems for us.
All right.
Okay, can we answer these questions? Some of them we have answered, but I just need to do it to be sure that we know what we are talking about. So for the first one, we say that we can measure the heat given off during the process of respiration. Okay, that's how we can measure, demonstrate that energy is given off. And then two, we talked about other forms of energy. Okay, these are not the only ones, but these are just a few. And then we can use the thermometer to measure the amount of heat given off, or the heat change when they are in the heat change when what? When respiration is taking place. Now, when you conduct that experiment, how would you do it? What are some of the materials you would need? And what are the sources of error? Maybe I can run us quickly. So this is how you would set up your experiment. So from here, you can notice that we have what we call a control experiment. We have the control experiment and the actual experiment. Like what we said last time, in science, it's always good to have a control experiment when you do an experiment. My question is, why do we need control experiments? Why do we need control experiments?
To avoid inconvenience.
Yes, Mirinda?
To give more information about the real experiment.
Yes, but the answer is not clear. It's not very clear. Patricia?
Okay, I think it's important to like have control experiments in order to ensure that the results which are observed are official, like they're not random.
Okay, they are not random. Daisy, what do you have to say?
Sir, according to me, I think why we have our control, it helps us to get accurate results in a real experiment. It helps us to get correct information, which is accurate.
You're correct, but your answer is not very clear. Dan Jackson?
It helps us to get correct information.
It's true you get correct information, but based on what? Why is it that we are able to get correct information?
It helps us to control and estimate.
Anyone? Mariam Grace? Alvega Mariam Grace? I think she forgot up. Please unmute.
Teacher, the control experiment helps us to know the fact that we are investigating the real experiment.
Okay, that is close enough. So when we conduct a control experiment and the actual experiment, it's very important to help us to verify whether our results we have got are truly as a result of the factor we are investigating, or they are caused by something else. That's why someone was saying that we want to be sure that we're not getting random results. Okay. So in that experiment we have just demonstrated, we have some sources of error. Now there could be heat loss to the surrounding. How is that a source of error? How can that result into a source of error, or and then how can we probably minimize that? Because if we go in front a bit, just a moment, sorry, you can see this is our setup. We have a vacuum flask. We have thermometer, we have boiled seeds, we have cotton wool. Now we are saying that as we set up this, we might have errors and our results may not be very accurate. So why would there be heat loss to the surrounding, and how can we deal with that? Because remember, when I don't know if we have ever had beans, say fresh beans, and you put them in a pan and you cover it, then in the morning you come and touch in the beans, do you notice that the temperature of those beans would be not the same as the room temperature? Have you ever noticed that? If you
Pardon, sir?
If you have fresh beans, assuming you bought or you got fresh beans from the garden, peeled them and put them in a saucepan or in any container, and then you cover that saucepan or that container. When you come in the morning, there are certain things you'll observe when you touch the beans. What are some of the things you might observe? Yes, Patricia.
Okay, sir. For me, it was not beans, but I observed with cassava. Okay, when we
Okay.
covered it, like we came back in the morning and it was, how do you say, it was warm.
It was warm. So it was warm, meaning that there was heat. But what else apart from the warmth? Let's have CJ. CJ, please talk to us.
Sir, I think I observed that drops on the container, inside the container.
Okay, so there was moisture. That was a correct observation. Okay. Now, in the same way, if you look at what I have here, just a moment. If you see what I have here, these seeds are expected to give off heat. And so when they give off heat, the heat is supposed to be measured by the thermometer that we have to be able to know which ones give off heat and which ones don't, or which ones give off much heat and which ones don't give off much heat, or which ones cause a reduction in the temperature. So there are three things that can happen when you're using a thermometer. The temperature may remain the same. That means the thermometer reading remains the same, or it may increase, or it may decrease. So if the thermometer increases, it means that there's heat loss. But if the thermometer decreases, means there's heat gain. And then if it remains the same, it means that there is neither heat gain nor heat loss by those seeds. So, why do we say there could be errors due to heat loss? And how can we sort that problem? We are dealing with a topic of respiration, and we are looking at the experiments to do with respiration, okay? And we're expected to be able to prove that heat is given off during respiration. So, but like we know, there may be sources of error. And one of the sources of error can be heat loss. So how do we minimize heat loss so that most of the heat generated remains in the flask without much of it going out and giving us wrong results? Remember, in this new curriculum, we have to do much of the thinking. For us, we are facilitators as teachers. I know the answers, but I would not help if I just told you what the answers are. So let's hear from CJ.
I think in order to control the heat loss, we use a vacuum flask because it prevents heat loss by either conduct.
Okay, I think we've lost CJ, but she was giving us good contributions. Rowan Mark, can you pick up from where she has left? Please unmute. Rowan Mark. Okay, let's hear from Mirinda.
In the chat,
Okay.
in the chat, somebody has said by placing the flasks in a water beaker, you minimize temperature fluctuations. Then another one has said by use of conductors of heat, and yeah, just that. That's all that is there.
Okay, thank you for your contributions. Lumu Joshua.
May I say that there are use of vacuum flasks that control heat loss by radiation, since they have a double silvery shining walls.
Okay, thank you all. Now, these people who are annotating, I'm going to exit you. Please, Hannah, can you exit these people who are doing this to us?
Okay, teacher, put them in the waiting room.
Yes, put them there until they learn what to do when they are. Okay, so your contributions are okay. So let us see what we have here. So possible source of error, we said heat loss to the surrounding. Now, what can cause heat loss to the surrounding may be when you don't use a vacuum flask, as Joshua has said. If you use a a conical flask, you see what we have number 5 here. For them, they have a single layer, so they easily conduct heat, either from the surrounding to the container or from the container into the surrounding. So that means your thermometer will not read all the heat lost. But also, if you don't use enough cotton wool, because if you look, we have cotton wool here. If the cotton wool is not well put, then there's going to be escape of heat to the surrounding, which is going to be a big issue. Okay, so that is very, very important. But if we go back, then the variation in seed mass. So for the control experiment and the actual experiment, you must have seeds of almost the same mass. So to avoid that, you have to do something. How can we avoid the variations in the seed mass? Because, you know, if one container has more seeds than the other, it means it will produce more heat than the other. So how can we avoid that source of error or that excuse? Someone may tell you, no, you put fewer seeds here and you put more seeds there. So how can we deal with that? How can we avoid the variation in seed mass? Because you are comparing. How can we avoid that? Do we have anything in the chat? Or do we have...
In the chat, somebody has said that by using insulators, by weighing, or then counting or measuring.
Okay, thank you. So weighing is a more accurate answer because counting them may not help. Some seeds may be, they may both be of the same number, but when some seeds are bigger, and so the weight will vary. So weighing them is the most appropriate thing. So you must weigh them and ensure that they have the same masses, okay? And even as you're reading your thermometer, you may read it inaccurately, and that can cause errors. Yes, Sheila? Sheila raised her hand. Please ask your question. Or raise your contribution. Okay, maybe we proceed. Come on, saying use the same number. I've just told you using the same number may not help. Weighing them is much better. So we've already dealt with number 5. And then number 6, you're asking why is the vacuum flask inverted? Why don't we have it standing upright like the way we have why don't we have it like the way we have a normal flask? Why do we do that? You notice the vacuum flasks are inverted here. Yes, Patricia?
I think maybe to prevent the heat from escaping, because I think the heat rises up. So if they were like that, they were up, let me say facing up here, the heat would escape, I think.
Thank you, Patricia. Well, it's not that, but thanks for trying. Lumu?
Yeah, I say that the flasks are inverted so that, okay, there's nothing, like no harm comes to the seeds, or to prevent them from getting contaminated or even being harmed. So that's why it is turned upside down.
Okay, thank you for trying. So what happens, I think someone posted something in the chat. Can we read what is in the chat, Hannah?
To prevent the seeds from coming in contact with the water.
Yes, so Joyce, why do we want to prevent them from coming in contact with water? What is wrong with that? So where does the water go to? And can we elaborate on that? Joyce, please explain your point. Joyce? Okay, let's have Daisy explain it.
So according to me, I think they are made upside down in order, because for example, when the seeds are, maybe when the seeds are in the like for example, when the seeds are upside down, it will be able for us to record the accurate temperature recordings, because they will all gather together. But when they are down, it will be hard for us to get the accurate temperature recordings.
Somewhat, although it's not very clear. It's not very clear. JK, do you want to build on what she has told us?
Okay, what I think is that the warm air rises up and the inverting will ensure that the heat will not escape through the cotton wool.
Not quite. So what happens is that when you put it upside down, you're preventing the seeds from coming in direct contact with the water. So the water drains out, okay? Because if the water remains there, it may cause the seeds maybe to rot, or the water may absorb some of the heat, okay? So those are some of the things, but also it is easier for you to get a more accurate reading because you pull the thermometer out slightly without it, because you know, if the, let me just illustrate my point. Let's annotate. Just a moment. So let's do this. So assuming you have your thing like this, your flask, like that. So then you have your thermometer inside there. So each time you pull it out, each time you pull out the thermometer, remember that seeds have remained at the bottom and here you have space. So the reading you'll take will not be for the seeds. It may be now for this space around there, because the seeds are the ones which are generating much of the heat. So, but when it's upside down, when you pull it out, even if the thermometer stops somewhere here, the bulb, it will mean that it's still reading the heat coming off from these seeds. So those are the two reasons why we do that. It is a lot easier to read this thermometer when it is like this, but also the water drains out. Okay, so that is why we don't put it that way. Okay, so let's proceed. Mirinda, you have a question?
Yeah, my question is, okay, like here in the answer, I'm saying something of water, but in the experimental setup, I'm not seeing any water. That's why. Where is the water coming from?
These seeds, when they respire, remember one of you told us that when they kept cassava, they noticed that in the morning it had sweated, for lack of a better word, okay? It had given enough heat. So during respiration, heat is generated and also water is also generated. So if each one of these seeds is giving off some water or some heat, some moisture, then the moisture will collect and drip out of this setup, okay? So that is why we must do that. Okay, so let's proceed. I have other questions. So why the control is necessary? Why were the seeds boiled? You noticed in the control the seeds were boiled and then later they were sterilized. Okay, so we have 5 other questions concerning this experiment. How can we answer these questions? The control experiment, why it's necessary, we already talked about that, okay? We said that to ensure that any temperature changes observed are due to the respiration of the seeds and not other factors, okay? But then why did we boil the seeds in one of them and didn't boil them in the other? I can see the chat is busy. Can we read from their contributions? So, Alan, please ask your question or make your contribution.
Okay, for me, what I think, I'm contributing about the seeds being boiled. So for me, what I think is because so that they do not germinate and utilize the oxygen or produce carbon dioxide gas.
Okay, you're correct, but it's not coming out well. Why don't you want them to germinate? Why don't you want them to germinate? We have Mirinda. Mirinda is quite busy today. Maybe is anyone who has not said anything this lesson?
Yeah, there is Alan.
Okay.
Alan, unmute.
Alan, please.
No, sorry. That was an accident. Sorry.
Okay. Okay, let's hear from Daisy. Oh, Becky, let's have Becky. Becky has not said anything.
I think in boiled seeds, like they have dead cells because they are killed during boiling.
So that is true. That's also a correct observation. So when we boil them, what are we trying to do? Daisy, can you harmonize what Becky has said?
Example, if we don't know this, leave them. When you see that trying to come out of the seeds, but if we boil them, we will kill the correct temperature recording from them.
Okay, so this is what I have. So we are denaturing the enzymes and stopping any metabolic processes, okay, before we start our experiment. Because remember, in one of the experiments, the seeds are live. That means they can carry out the process of respiration. But if we boil them, then they become dead seeds. That means they can't carry out respiration. And so it means we expect the dead seeds not to produce heat since they are dead, because dead things don't respire. And so they don't generate heat, okay? And then we expect the live seeds to generate heat from the process of respiration, because we know that living things respire. So if you want to do a good experiment, you must have a control whereby you can compare the results and know that the actual outcome of your experiment is due to that factor you are investigating, which in this case is the factor of respiration being able to produce heat, and which heat is seen from the temperature change. So I hope that is very clear. But then, why do we sterilize them? Why don't we just boil them, cool them, and then just add them into the experiment? So what is sterilization and how is the sterilization done? And rather, why is it done? Yes, Daisy?
Sir, I think sterilization is getting those seeds, then place them in water, and I think that is done. For example, if you place the seeds in water, that water will get spice in the seed and check them maybe like the following day. You see that they will be more soft and they will be somehow like the temperature will increase them.
Okay, Daisy, I think I heard most of what you said, but if your network is not good, this goes to everyone. If you realize your network's not good, please just type. The message will come. Talking may not allow us to hear you very well. Alani?
Okay, I think they sterilize the seeds to eliminate microorganisms which may interfere with the experiment.
So that is correct, but then now how do we sterilize? That is why we sterilize, but how do we sterilize? What do we use? What are some of the options if we want to sterilize them? Lani? So when we are sterilizing, we are basically trying to kill the microorganisms. You know, when those boiled seeds remain.
By chemical treatment.
Yes, what chemicals? What I want to have an idea about. Let's hear from Stace Pop 5.
So by boiling it.
Boiling will not sterilize, it will only kill them. Bridget? So we are going to use a chemical like Alani said, but we are going to use either formalin. Formalin is used to treat dead bodies so that they don't smell, okay? Or we can use hypochlorite. So all those things are able to kill the microorganisms, because if the microorganisms start feasting on those boiled beans, they will respire and also generate heat, and the experiment will no longer make sense. Okay, let's move on. So to eliminate microorganisms that might interfere with results, then why are the seeds soaked? Why are they pre-soaked and then not packed in the flasks? We notice that the time frame, of course, is 24 hours or 1 day. Yes, Ryan?
Sir, my question is on the answer of 3. I thought boiling also kills the microorganisms.
It might kill them, but remember, microorganisms are found everywhere. Even in the air we breathe, there are microorganisms, the spores, fungal spores and what have you. When something cools down, they fall on it and then start decomposing it. So boiling is not enough. When you put that chemical treatment like formalin, when they fall in there, they are killed. Hope that is clear. Okay, so why are the seeds soaked? Pre-soaked. So here and then, why are they not packed in the flask? We noticed there was space between the seeds and the flask. Lani?
Okay, for me, I know why they are soaked.
Okay.
To ensure that they are hydrated and viable.
Okay, so it's correct, but I want you to give me more. I think some people may not understand it the way you understand it. Can you tell us a little more about that? Hydrated and viable. Viable for what? What's the importance of that water?
It helps in promoting active metabolic processes.
Which particular process is that you're dodging?
Respiration.
Respiration, but most importantly, germination, because respiration and germination, during germination, respiration is taking place, okay?
Okay,
Energy
sir.
is utilized in causing the plant to grow. All right, and then lastly, on this same question, we see the packing. So we don't pack them because this is going to allow for oxygen supply for the seeds to respire, okay? So what happens is that when you pack them, then there's little oxygen available, and therefore less respiration will occur. So we'll have anaerobic respiration, which doesn't generate much heat. And it may take our experiment longer for us to realize any serious results. Okay. Let's go on to the next experiment. Here we have another experiment to show that production of carbon dioxide happens during respiration.
Teacher, we are not yet done.
Sorry?
Teacher, we're not yet done with the previous screen, just a minute.
Okay, let me go back just a minute.
Thank you.
There are some words that were cut off here, so you may not see them, so you just have to follow what I said. Oh.
Daddy, a baby. He's a baby.
So I hope we are done with that so that we can move on.
Thank you.
So I can continue.
Okay. So here we have another experiment in which we want to show that there's production of carbon dioxide when our respiration takes. Okay. So what is the purpose of the sodium hydroxide? Now, before I go any further, in this experiment, like we saw, you must have a title. Then you must have a hypothesis, which would say carbon dioxide is produced during respiration or carbon dioxide is not produced during respiration. Then the materials are everything you see inside here. Then the procedure is how you set up this thing. And then the observations are what we are going to mostly discuss. Lani?
Okay, like from what I know is sodium hydroxide is used to absorb carbon dioxide during the experiment. Remember, like sodium hydroxide absorbs carbon dioxide, so that is the use.
Okay, so it will absorb carbon dioxide.
Yes, sir.
Before I forget, what is at point X here where my cursor is? If you want this experiment to work out, what must you put here?
A suction pump.
Okay, you must put a suction pump. What's the importance of the suction pump? What does sucking mean? Yes?
Direct air.
The pump keeps the process moving in one direction.
What do you mean by the process? Be very clear. What in the process are you interested in to be moving in one direction?
Like the gases.
One person at a time?
Sorry.
Yes? Was it Gabriel who was talking?
Yeah, it was. It is.
Yes.
From the air, the gases. To keep the gases moving in one direction.
Okay, yes. So you want air to move in through this point and come out through this point. So you want to have a continuous stream of air moving in the setup and exiting at this point. So that is very correct. So why do we have lime water A and lime water B? Why do we have lime water A and lime water B? Why do you have lime water A and lime water B? I'm seeing the same people all the time, yet I'm having an attendance of 92, 93. So I expect as many of us to participate, either in the chat or elsewhere. My monitress should be reading the chat. Pick out only the answers relevant to our discussion. It seems nobody has answered the question at hand. Why do we need lime water A and lime water B? Let's have Jolene, I think.
I think the lime water is used to test if there is carbon dioxide, because if there's of carbon dioxide, the lime water turns milky. So maybe the lime water is there to test if there is carbon dioxide.
Okay. Now, but why do we have two lime waters? We have lime water A and lime water B. Your answer is partly correct. Nairobi, there's someone who had the Nairobi. Nairobi, can you answer?
For lime water to test that plants produce carbon dioxide when they are test that carbon dioxide is produced when animals are respiring.
Lime water A? I think something. Let's hear from Peach. Because we are not having plants, in this experiment we have animals. Peach, please unmute. Okay, let's hear from Patricia.
Okay, thank you, sir. Well, I think the lime water in A is used to confirm the absence of carbon dioxide in the incoming air, because remember, it first went through the sodium hydroxide like the air. So you have to confirm if the carbon dioxide was absorbed. So that's what I think.
Yes, so you're confirming absence from the incoming air because we expect to have all been absorbed by sodium hydroxide. Okay, what about lime water B? Lime water B, let's hear from Daisy. Let's hear from Daisy. Daisy, please post in the chat. If we cannot hear you, you can as well post your answer in the chat. Okay, let's hear from Alan. Or let's hear from Zanita.
Good afternoon.
Good afternoon.
Lime water B is to confirm that carbon dioxide is produced since the animal produces carbon dioxide after like respiration.
Okay. All right. So therefore, if you know that is the purpose, what are the expected observations in lime water A and lime water B? I prefer somebody who has not contributed to give others chance as well.
In the chat.
Yes, in the chat.
Somebody has said that lime water B absorbs and proves that the animal is producing carbon dioxide by turning milky. Then that lime water B is to confirm whether animals produce carbon dioxide during respiration, since we believe that air from air is carbon dioxide. Yeah, that is in the chat.
Okay, the chat has only told us about lime water B, that it will turn milky, which is true. But what about lime water A? What will be observed on the lime water A? Let's give Peach another chance. Peach. Okay, let's hear from Shannon.
I think lime water air will remain the same because there is no carbon.
I can't hear you, Shannon. Maybe you can post in the chat.
Okay.
Let's hear from Bridget.
So I think lime water air will remain the same since there will be no carbon dioxide.
Okay, by remaining the same, what do you mean? Do we all know what the color of lime water is? So we need a
Teacher, I'm
clearer
not sure
answer.
about the color of the lime water, but the color won't change. It will remain the same.
Alan, what do you have to say about that?
Okay, I think that the lime water will remain colorless.
Alan, you're not very clear.
I think that the lime water will remain colorless.
Okay. Yes, so the lime water remains colorless. In other words, its color doesn't change from colorless to anything else. So when I talk about changes of lime water, it's always from colorless to milky. All right, I think we've answered those questions. So if we wanted to do the same experiment, but this time using a plant, what should we do to make this experiment work for us? What should we do? Okay, Lani, that is right. Daisy, that's correct. I'm seeing sample in the chat. Okay, the question is on the screen. Question number 3. What would you do to ensure that you can still do a similar experiment, but this time using plants and not animals, as we have done? Yes, Patricia. Can we describe what we could do to modify this experiment? I see the chat is a bit busy. Now, some of you are busy chatting about other things. May be forced to be removed from the discussion.
Teacher, the last message I saw was that it remains colorless.
No, that question we have answered, and now we are dealing with question number 3.
All right.
Yes.
But that was.
So in question number 3, all we would need to do is simply to put a plant here where the mouse is, and then cover this bell jar with the black paper, or cover it with something that is opaque. Now the question is, why do we cover the bell jar with an opaque substance? If we are going to put a plant there, why don't we just put a plant there and conduct our experiment? Why do we have to do that? So Let's hear from Alan, then Daisy, then Patricia.
Okay, for me, what I think is that to prevent light from coming into the jar and preventing the plant from carrying out photosynthesis.
Okay. Daisy, why don't we want photosynthesis to happen?
Because we don't want photosynthesis to happen because there will be manufacturing of food.
What's wrong with manufacturing of food? Is that a problem? There's something you're dodging that I want you to say. Patricia, then from Patricia, we go to Rowan.
Okay.
Then we shall go to CJ, and then we shall go to Samsung, in that order.
All right, I think we don't want the photosynthesis to go on because here, like the carbon dioxide is going to be, how do you say, used and oxygen will be released. Yet we are trying to experiment if carbon dioxide is present. I mean, we are trying to experiment the production of carbon dioxide during respiration. So in case we leave the light there, as in we don't cover that tube or flask, then the light is going to affect the plant. It will carry out photosynthesis, and then the carbon dioxide which we needed will not be produced. Instead, it will be oxygen.
Okay, thank you, Patricia. Does anyone have anything more to add on Patricia's contribution? CJ's hand has remained up, and Rowan. In the same order I had told you to respond. I'm sorry, I didn't hear that.
To prevent interference between the experiments.
Not very clear. CJ?
Sir, I think that during photosynthesis, carbon dioxide is not given, but instead oxygen. So we cannot get the correct results because carbon dioxide will not be produced.
Sorry?
Carbon dioxide is not.
Carbon dioxide is not what? Okay, so the answer is basically what Patricia said. Patricia?
Yes.
Patricia gave a correct answer. Because if light is there, then photosynthesis will take place. And when photosynthesis takes place, it uses the carbon dioxide. And therefore, there will be no carbon dioxide available to be detected in this flask. Okay. Okay, so you will not detect anything. So that's what we must cover to prevent photosynthesis, and therefore, so that none of the carbon dioxide produced by respiration is used. And wash them first. Yes, Lani.
So which number are we on? Like, I'm confused. I think I had gone somewhere.
Okay, we've just finished number 3. Now we are looking at number 4.
Okay.
So if we are to look at a suitable control experiment, what would it be like? What would a suitable experiment be like for this case? I'm seeing a number of people whose hands are up. I'll give those who have not said much first. Let's have Samsung SM. Then we shall have Techno Spark. Oh, Techno has run away. Samsung, unmute and give us an answer. Then we shall have Nairobi, Mirinda, and Daisy. In that order.
Teacher, good afternoon.
Good afternoon.
So may I think that in our control experiment, instead of using sodium hydroxide, which absorbs the carbon dioxide, we shall have to use sodium carbonate. That's what I think.
Not really. Nairobi. Nairobi next.
So there on the mouse, we shall use a dead one.
A dead one. Why a dead one?
A dead mouse. Because now a dead mouse will not respire and the carbon dioxide will not be produced.
Yes and no. In principle, what you're saying is correct, but in practical terms, it may not bring the result. I don't know if anyone can explain.
Now we shall, in actual sense, we shall use the dead one.
Yeah, I've just told you that the dead one will not bring the result we want. But
okay.
Yes. What do we know about dead organisms?
They don't respire.
They don't respire, yes, but what more do we know about them? I think your friends want to respond.
Okay.
Let's hear from Daisy, then Tasha.
So let me first respond from what he's saying. We can't use a dead mouse because remember, a dead mouse can't react to anything. It will be as useless as nothing. So it won't help in that experiment. And our control experiment, which I was suggesting, which we can use is by trying to remove B, lime water A.
Not really. We don't have to do anything with the lime water. Tasha?
Okay, so for me, I think we should use the same equipment, like the everything, the conditions as the experiment itself. Then after in the control experiment, you exclude the living organism from the control experiment. Then you maintain the same temperature, pressure, and other environmental factors in both the setups, like the experiment and the control experiment.
Okay, that is correct. Tasha and Nairobi have similar answers, but I may go with Tasha's answer. Why? Let me explain. Because we know most of these animals, when they die, they start decomposing, have microorganisms in them, okay? So you would rather, unless you're going to get the organism, kill it and sterilize it, okay? But if this organism is dead and not sterilized, the microorganisms inside there may respire and give us similar results. So that's why you either leave the flask empty or you get beads or anything that is non-living, like beads, you can get stones or something, and you put it there. After, of course, you've washed them and dried them, okay? Then you would get your control. But if you just get an animal you have just killed and then put it there, then remember in our bodies we have microorganisms. They may start the process of decomposition, which in itself produces carbon dioxide. So that's why I said his answer is correct, at the same time not so correct. So you have to be very careful how you design your control. In principle, you put something that is non-living. So a dead thing is non-living, but it may be dead yet carrying microorganisms which could still respire. Nairobi, you want to ask something? Okay, I think they forgot their hand up. So it is fine. Let us continue. Let us continue. Our time is fast running out. But
Yes,
I hope people have understood. Yes, Lumu Joshua.
I have a question. Okay, we have discussed like this slide, but then how we get answers because
I'm not hearing you. Maybe you type in the chat. Amara Mwezi.
Teacher, sorry. Teacher, I was asking, now, as you are saying, observation, number 2, describe the expected observation in lime water, A and B. Because as you're saying that now with the sodium hydroxide will like trap.
Sorry, what do you ask about sodium hydroxide?
No, I'm saying I want to understand something with the purpose of sodium hydroxide and lime water B, because we are trying to show carbon dioxide is produced by respiration, right? But what's confusing me is that now as the carbon dioxide is passing through, what will the sodium hydroxide really, how would I say, contribute, and what will happen to lime water B, since the carbon dioxide already passed through lime water A?
Okay, I think CJ maybe wants to answer you. If he doesn't, then I'll answer you. CJ, please respond to him.
Sodium hydroxide absorbs the atmosphere so that it's only oxygen that's going to pass through to the animal. Then it gives off the carbon dioxide, and that's where lime water B will turn white to show that carbon dioxide was produced.
So I hope, Muhwezi, you've understood.
Yes, but teacher, now I'm asking, now what will happen to lime water? Will it also turn a milky color or it won't?
Which lime water?
I'm saying now between lime water A and B. I've understood sodium hydroxide, but I'm saying between that two, will they both turn milky or one has to turn milky?
It's not like one has to turn. It's the question is, which one will turn and why will it turn? If you have followed CJ's explanation, lime water A shouldn't turn if you have enough sodium hydroxide to absorb all the carbon dioxide. But if lime water A turns, it means that some carbon dioxide has managed to come in here. So if some carbon dioxide comes in here, it means that you have no guarantee that carbon dioxide hasn't come here. Okay? And if carbon dioxide comes right here, then you cannot be sure that it's not the carbon dioxide which has caused this lime water to turn milky. So you want to be sure that the source of milkiness in this lime water B is only from the animal. Since the animal did not, the carbon dioxide that came from the atmosphere was all absorbed by the sodium hydroxide. So that is the purpose. That's the idea behind it all. Okay?
Okay.
It's the idea. So if your experiment doesn't produce something like that, it means that there's something you did wrong. Either you have low concentration of sodium hydroxide, or the sodium hydroxide is of little quantity, and that is where the problem might be. Okay.
So, teacher, in this main experiment, respiration,
Yes.
the way the production of respiration is supposed to be helping the production of carbon
Can
dioxide.
you increase the volume of your voice? I can't hear what you're saying.
Which I'm saying, like, as you have said, yes, that now this carbon dioxide is being produced by respiration. So as the animals respiring, carbon dioxide is being produced, timing lime water B, milky, right? Then with the sodium hydroxide, it will absorb most of the carbon dioxide, hence lime water A won't turn milky. Is that's what I've gotten? Is that what you're trying to mean? This?
Yes, because at the beginning of this discussion, I told you that at point X where my cursor is, we have a suction pump. Means that air flows only in this direction. It cannot go backwards and turn this lime water. Yes. So the air only flows in this direction. So whatever carbon dioxide is getting off will only come and turn this one milky. It can't go and turn this one. Okay. That's a very important thing to take note of. Because if you have no suction pump here, then the carbon dioxide is free to go wherever it chooses.
Yes.
And in that case, it means that this can turn milky and this can turn. So those are also sources of error. Failure to put a suction pump. Okay. The other source of error can be if you don't make sure that this inlet is touching the sodium hydroxide and this one is touching the lime water. You notice that the outlets are always up and the inlets are always down. Okay. In fact, in some books, this one would be further down up to somewhere here. Okay. To ensure that the oxygen is close enough to the animal. And then the carbon dioxide given off rises because warm air rises. So it will rise and then come and turn this lime water milky.
Okay, teacher, thank you so much.
Okay, so I think we have done that. We have a few minutes. Maybe we can squeeze in another experiment and then call it a day. Yes, Joshua.
Can you hear me very well?
Not so well, but you can just change your position. Joshua, maybe you put your question in the chat or your contribution.
It seems it has been responded.
Put it in the chat. Then we have a question here. Okay. So this is also another question. What is the purpose of this experiment? Notice that here we have sodium bicarbonate, we have hydrogen carbonate or sodium bicarbonate indicator. We have soap seeds which are germinating. After some time, the bicarbonate indicator changes this color. Then here where we have dead seeds, the bicarbonate indicator did not change. So this is your homework. Maybe you can go and take a screenshot and go and try out those. There's someone who's Narubeg, I'm going to exit you. So let me allow you to make screenshots, then you try those things out. Okay.
So are we trying them out in our free time or from here?
Probably in your free time.
Okay.
Okay. I wanted to.
Excuse me, sir.
Yes.
I think Joshua has a question in the chat, but no one is seeing it.
Oh. So Joshua, what's your question? I have a question in the chat, but not being attended. What question is that? I have not seen any question. I had someone to attend to that question, to bring attention questions to my attention, but nobody has. Maybe you type it again, copy it and paste it.
Sir, the question is, for this is there a slide that is having answer?
No, the answers, we have discussed them. So you need to have been attentive.
So he wasn't in the lesson. So like he had gone out. That's why. I think he literally wants you to go back like some previous one.
Yes.
It will take us a lot of time because people keep coming in and going out, okay? I don't have the answers, but I discussed those answers.
You can view the recording on YouTube.
Yes, there will be a recording on YouTube, so you can go and do a review. Thank you. So let's have Patricia.
Okay, sir. Okay, these experiments you have been carrying out, is it like to prove respiration in general or a type of respiration?
Because experiments are about respiration. Now the question is, okay, what you need to understand also is that all experiments, we don't have one type of experiment. Like I began the lesson, I said, the experiment can be conducted based on the resources you have at your disposal. So if you had sodium bicarbonate and you don't have lime water, you can still conduct an experiment, okay? And so I don't want to give out the answers because I'll be robbing you of the chance to learn. So I want you to do research on that. Just photo it. You can do research online. You can do research from the textbooks and then see how you might answer those questions, okay?
Okay.
Yeah, remember in our new curriculum, which is typical of life, you don't get all the answers, okay? I could give the answers, but I don't want to. Tasha?
Okay, sir, like on your screen, I'm seeing observing water release in inspiring seed.
Yes.
Inspiring, is it also something we have to research about?
I thought that in the remaining time we can run over it.
Okay, sir.
Yes, but before we run over it, I think you have ideas. Assuming someone told you that they are going to design an experiment that wants to show that actually water is given off by respiring seeds, how would you advise them to plan that experiment or to design the experiment? What materials might you suggest to them to use? Yes, we have Tasha, then Mirinda. What would you suggest to them? Tasha?
My friend.
Okay, Mirinda.
So for me, what I would advise them is that they can have some conical flasks, delivery tubes, and the animal or plant. But in general, they must not miss out is that they must have anhydrous copper 2 sulfate. Or you can use cobalt 2 chloride paper. Yeah.
Okay, thank you for your contribution. I think now Tasha has something to say.
Yeah, like my phone had frozen, so now I've come back. Me, what I would tell them is, first and foremost, they need like organism, like a plant, like plant seeds. Then an airtight container, then a desiccant, like sodium hydroxide, a substance that absorbs, no, not sodium hydroxide, like a substance that absorbs water, then rubber. Yeah.
Okay, thank you, Tasha. Patricia, you want to say something?
It's almost similar to hers, but I think they'll need like seeds because these give out moisture and maybe they may need a delivery tube, I think.
Okay, I can hear answers here and there. It's unfortunate that we cannot see the setup, but I don't have the setup here per se, but we shall see how we can create one basing on what we have. So for materials, of course, we need germinating seeds. So you can use any seeds of your choice. Then you need two small airtight containers, okay? So why two? Because you need a control. One of them will serve as a control, okay? And they should be clear, ones where you can see through them, okay? Then cotton balls or absorbent material, something that will absorb the moisture, probably some water for soaking the seeds, a scale, plastic wrap or cling film, rubber bands, marker. So the procedure here, we start germinating the seeds by soaking them in water for a day or two. And then they begin to sprout, because when germination is taking place means the seeds are actually respiring. That's the whole idea. When they respire, it means they are going to give off water. And then weigh a certain amount of germinating seeds using a scale and record their initial weight. Then place a damp cloth or absorbent material at the bottom of each airtight container. Place a known quantity of germinating seeds on top of the bone cotton wool in each container. Okay. Now seal one container with this lid tightly. For the other container, cover it with a plastic wrap or cling film. Okay. Then label the containers to distinguish them between the sealed and unsealed ones. So then put them in a well-lit area. Then observe inside the containers over a course of several hours or maybe a day. Then the results are that in the unsealed container, you are going to see droplets, okay, or condensation forming on the sides. Then the sealed container may not show significant condensation, okay. Of course, I would rather modify this. I could even use dead seeds, okay? Dead seeds, that is you have boiled them, dried them, boiled them and then put them in a chemical to kill the microbes and then do the same setup, okay? And then you watch for condensation. So the conclusion is that germinating seeds release water vapor during the process of respiration, and the condensation in the unsealed container would be evidence that water is given off by the respiring seeds. This is another experiment, but time doesn't allow us to go any further. I think we'll leave it at this. Yes, let's hear from Razia.
So just step back, I want to take screenshots from where material is full.
Okay, Tasha, what is it?
Okay, so like when we complete doing the activity you've given us, where can we put like?
You discuss it with your teachers.
Okay,
Because
at
I don't
school.
think we are going, yes, I don't think we are going to meet next week, are we?
I don't know. We shall ask Mr. Kaziwa.
So we are meeting for next week, Monday is 22nd. So we shall meet.
We shall meet. Okay, if
Yeah,
we are meeting, then you will share the ideas, the answers with me.
Okay.
Okay, sir.
Okay. So let me go back for people to take a few screenshots. Okay, so there's something that you may need to modify in this, in the sense that you may need to work with germinating seeds and then seeds which have been boiled and sterilized in the other container. Okay, so that is very important adjustment. I think I need to correct that somewhere. Then this is the second slide. Remember, the weighing is to ensure that you have the same quantity of seeds in terms of amount of water likely to be produced.
I can proceed to the next slide.
Okay. So with that, I think we need to close off this discussion. I will request someone to lead us in prayer. Any volunteers?
Let's sum ourselves a word of prayer. Almighty Father, King of Kings, we want to thank you for enabling us to come for this lesson, Father, King of Kings. As you're going out of the lesson, may you enable us to go when we have all at least had something we have kept in our brains, Almighty Father, King of Kings. We know we may not be as perfect as someone may think, Father, King of Kings, but you know we are perfect in your eyes, Father, King of Kings. May you continue to bless us in every step that you make, Father, King of Kings, because I believe one by one makes a band, Father, King of Kings. Father, may you continue blessing our teacher and enable him to also continue teaching more people so that they can be as prosperous as him. Future biologists come, Father, King of Kings, pharmacists, Father, King of Kings, surgeons, doctors, and whatsoever people want to be. May you bless them. I've prayed, believing and trusting in the mighty name of Jesus Christ, our Lord. Amen.
Amen.
And thank you.
Amen.
Zanzi, come go to my DM, I mean on WhatsApp.
Natasha.
Then Quarin Agatha, come to my DM.
Hi everyone. Back.
Gen Z, I'm coming. As in, okay, I'm coming.
Gen Z, just now I'm waiting. And by the way, Patricia, you didn't complete explaining to me the chemistry work.
I'm coming.
Okay.
It's gone off.
Other guys, bye. We shall meet if the coming lesson.
Bye.
Yeah,
Bye.
guys, bye.
I hope you